Hello and welcome to my talk. My name is Tvrdik and I'm excited that the work from my lab has been selected for virtual presentation. This is an early stage project titled Limiting Clonal Expansion of Cerebral Cavernous Malformations using Sonodynamic Therapy with Focus Ultrasound. It is well established that cerebral cavernous malformations, CCM, are caused by heterozygous germline mutations in one of three genes, CRIT1, CCM2, or PDCD10. Even the sporadic form, the somatic mutations are occurring in the same CCM genes. The familial form presents with multiple brain lesions that are best imaged with T2-weighted MRI. The different flavors of T2 have different sensitivity. For example, the T2 star and GRE detects more lesions, and the susceptibility weighted imaging method has had the highest sensitivity. However, this approach doesn't work very well in the rodent model. CCM mutations cause aberrant signaling in multiple pathways. Can be roughly divided in two areas. One is more acute and structure-related and affects, for example, the tight junctions between endothelial cells. The other is more adaptive or chronic and acts through transcription factors that are dysregulated in this disease. KLF2 and KLF4 are two transcription factors that are particularly important in this malady, as they are involved in the process known as endothelial to mesenchymal transition, NMT. So this mechanism contributes to a number of pathologies, but in blood vessels it causes the elongated endothelial cells to become more round, develop short processes, and differentiate into pericytes, fibroblasts, smooth muscle cells, and other cell types. So this process causes blood vessel thickening and other problems. Recently, two publications shed light on the mechanism of formation of the CCM. One paper by Malinverno and others from Dejana group in Italy, and the other publication by Dether and others from the Marchuk lab. So these are two different artistic renditions of the same model, and both group arrived at the conclusion that the small size lesions up to approximately 100 micron in diameter are clonally derived from a single mutant cell in the blood and blood vessel. After that, however, the mutant cells caused de-differentiation of the neighboring endothelial cell in the blood vessel and recruit them in the malformation, enlarging the size and becoming very heterogeneous, prone to bleeding. And these large lesions are very difficult to remove surgically, particularly when seated deeply in the brainstem, for example. So alternative approaches are needed. With collaborators, we put forward a proposal that the use of focus ultrasound could perhaps control and reduce the burden of CCM. So we use a focus ultrasound system adapted for small rodents, for mice, as shown in this diagram. It consists of the pattern generator and amplifier that controls the transducer, which generates the focused beam of ultrasound energy that can be focused in a small spot in the mouse brain. The mouse head is shaved, but other than that, intact. And if coupled with MRI imaging equipment, the the focal point of the ultrasound energy could be precisely controlled and monitored. So high energy focus ultrasound is often used as incision-free surgery, for example, in patients with Parkinson's disease. But in this publication from Richard Price lab at the University of Virginia, it is shown that a low energy ultrasound might have certain advantages. It's still capable of manipulating endothelial cells in blood vessels, for example, with the use of microbubbles, but it does not cause blood-brain barrier disruption or inflammation. And this is really important for our project idea because inflammation or blood-brain disruption would be really detrimental in the treatment of CCM. Another study from University of Virginia by Dr. Prada and others shows that certain chemicals known as sonosensitizers can produce reactive oxygen species when exposed to ultrasound energy and cause cell death in nearby cells and tissue. So as shown in this study, fluorescein, when delivered to glioma in the rat model, could lead to ablation of the tumor cells in the combination of with focus ultrasound, and reduce the total tumor volume as shown in this study. This is important for our hypothesis because if we deliver fluorescein specifically to CCM, we could perhaps use low energy ultrasound to ablate CCM mutant cells without disrupting blood-brain barrier. So here is our experimental design. We use the conditional fluxed mouse lines developed by Dr. Whitehead and Lee at the University of Utah. We induce the lesions. We induce the deletions with PDGFB, CREOR mouse tool previously published, and we also include in this strategy a fluorescent protein reporter that we developed ourselves several years ago. We induced the mutation in the mouse model with low concentration tamoxifen on postnatal day 5, and we have characterized the first round of these mutants with MRI and histology. We have found that the T2 space sequence and on our 7 Tesla machine at the University of Utah and University of Virginia detects the lesions with high sensitivity and precision, and we've done correlation with histological staining with neural trace NISL and isolectin IB4 labeled with fluorescent dyes. And we show that these lesions are distributed throughout the whole brain, here in subcortical areas near the lateral ventricle, or here near the dorsal hippocampus. In all cases, the lesions showed very high affinity for isolectin. So these findings led us to refine our initial hypothesis, and we proposed that isolectin could be perhaps used to specifically deliver the sonosensitizer fluorescein in the CCM lesions. And after MRI-guided FUS, we will be able to gently ablate these mutant CCM cells and perhaps ultimately limit growth and clonal expansion of CCM lesions. So in conclusion, we have established a chronic mouse model of CRIT1 CCM pathology, and which we established by using tamoxifen on postnatal day 5, and we work on the CCM2 model in parallel. We have observed high incidence of large lesions throughout the whole brain. And histological staining indicated that these lesions have high affinity for isolectin GSIB4. And so in our next step, we will infuse intravenously these mutants with fluorescently labeled, FITC labeled isolectin IB4, and we'll use MRI-guided FUS to expose these animals specifically in the lesions, and we will monitor the size of the lesions over time. We will also use another sonosensitizer known as 5-ALA. 5-ALA is an FDA-approved dye that is used in photodynamic detection and surgery of cancer, and we will compare these two approaches. And finally, by the use of the fluorescently labeled reporter, we will be able to determine if CRIT1 and CCM-based mutants and lesions expand clonally, as similar to the PDCD10 model that was published recently. So this work started in my lab at the University of Virginia by postdoctoral fellow Khadija Sharifi, and it is done in close collaboration with the Price Lab at UVA. Two graduate students, Katie Gorick and Delaney Fisher, who will be taking care of the next step in this project in collaboration with G.N. Kumar, who is the neurosurgery resident at UVA. And we are funded by the Focus Ultrasound Foundation. And thank you for your attention. If you have any questions or comments, please email me at tvrdik@virginia.edu or give me a call on the phone number below. Thank you.